Thank you. And I'd like to thank the organizers for inviting me to be part of the symposium. I'm mindful of the fact that I'm the last presentation before the break, so I'm going to be efficient as I move through the slides. So I'm going to be talking about cold plasma, which is kind of a new technology that is still very much in the R&D phase. And there will be some similarities, some analogies to what has been presented in the previous speakers about what you have to do to get this validated and properly understood as you're moving it to the point of being useful for the industry. So just kind of a quick outline of what I'm going to be talking about is some of the stages of where we are in terms of the R&D for cold plasma and where it comes from, how it's an adaptation of some existing technologies, and what you need to do to move towards these optimized protocols that will allow you to use it responsibly and reliably. What you have to do to validate this, to make it effective and predictable and reliable in using it. And that's going to talk a little bit about the underlying mechanisms and modes of action for antimicrobial efficacy. And then just talk about some of the pitfalls and some of the things that are associated with cold plasma, which can make it a little bit tricky for validation, at least at this stage of technology development. Again, going to some of the modes of action and some of the operational variables associated with the technology. So first, let me just explain what cold plasma is. For those of you who are here for the first talks and now you're into something that you maybe don't know what it is. So as we all remember from our physics classes, there are three states of matter, right? There's solids, liquids, and gases. Well, plasma is the fourth state of matter. And as the diagram shows, as you add energy to a solid, you break down the interactions between the atoms and molecules that make up that solid, and you lose that structure and you get free, moving independent molecules. If you keep adding energy to it, then those atoms and molecules bounce free of each other and they float individually and you've got a gas. Well, if you keep adding energy to it, you break down the structure within those atoms and molecules and you get free electrons floating around, you get ions, you get monoatomic oxygens, you get other broken particles that then create a plasma. All right. Some of the plasmas that we're probably familiar with are things like electric arcs, which we heard discussion of it in the RF technology. An electric arc is a tight, hot, dense plasma. Now the trick with cold plasma is to take that tight, hot, dense plasma and spread it out. So you get adiabatic expansion, so you get a loose, cool, diffuse plasma that still has all of these reactive products that are very good at killing bacteria. Right, and killing other organisms. Some other examples of plasma that you're probably familiar with, thermal plasmas, would be, say, the sun, candle flames, interstellar gas, plasma arc lamps, and things like that. Those aren't useful for killing bacteria, but some of the technologies that are useful in those can be adapted to create the kinds of plasmas that we can use to apply to foods and food systems. So the inputs to the system here are first energy. When I talked about you had to have some kind of energy to break down these structures, that could be in the form most frequently of a high voltage electricity, which is what we primarily use in my laboratory. But you can also use microwaves, you can use pumped lasers, and you can use some other things. And you need something to turn into a plasma. You need some kind of gas. We use air for the most part because it's around and it's cheap. It's got other limitations that I'll go into a little bit, but you can also use pure gases. One of the diagram that Dr. Anderson showed used an argon-based plasma. Argon's a great gas, it ionizes pretty readily. It's got advantages and disadvantages. You can use other gas mixtures, and I'll talk about that a little bit as we talk about other inputs. So what do you get when you make plasma? Well, you get what's called the self-quenching plasma, which means that if you don't do anything with the plasma, if it doesn't go onto the food, it doesn't interact with the food, eventually all the energy that you put into it, into breaking those things apart, will just kind of go away as they come back together. And it's so self-quenching. So you don't have a lot of residues that are coming out of this. You don't have any of these other products. Some of the longest lived products that are coming out of this would be, say, ozone or nitrous oxide produced in the cracking and reforming of the gas. And so chemical residues associated with the plasma are expected to be pretty minimal because you're not, creating any other kinds of halomethanes or, there's no chlorine that's added to the system or anything like that. This is a pretty new technology. There are no, to my knowledge, regulations on the books yet in terms of how it would be used for food processing. But these are the kinds of things that will be coming along as you're talking about taking it through validation to be useful in the real world. So let's talk about terminology. So when you say cold plasma, what exactly are you talking about? This is one of the big limitations with cold plasma, is there are lots of ways to make plasma. There are lots of different kinds of technologies and lots of different things that can go into this. Some of these can make plasmas which actually end up being rather hot, okay? And they're still called non-thermal because of the relative ratios of energies of the electrons versus the ionized particles. That's a very physics-based definition. Typically, when we're talking about cold plasmas or cool plasma, we're talking about things that are operating in or around room temperature. Now, that doesn't necessarily mean that they don't add any heat, but it means that they're operating in that kind of a temperature regime. Specifically, non-thermal is going to mean that it kills by some mechanism other than thermal processing, all right? There's a chemical interaction or some other kind of mode of action that's not based on heat. As the technology evolves, the nomenclature is going to settle down. Right now, as I said, there's many, many different kinds of designs of technologies with different electrode configurations and other things. Some of the naming conventions are based on the technology. So you'll have people refer to a gliding arc plasma or a plasma jet system or a corona discharge plasma system. These are based on the types of electrodes that you're using in that kind of a system. Others will talk about a helium oxygen plasma or an argon oxygen plasma that's based on the feed gas that you're using. Others of them will talk about that based on the applications. It can make it very difficult not only to compare one study to another, it can also make it a little bit tricky when you work with cold plasma and you're doing literature searches. So feel for me, guys. These different plasmas can have the same technology used by different laboratories. They might call it different things, which also makes for kind of a tricky situation. So the term cold plasma really is a loose term that covers a whole constellation of different kinds of technologies. A couple of real quick sketchy examples here. One, you can generate plasmas using radio frequency discharges inside a tube, which can be either evacuated or at 1 atmosphere. Rapidly cycling electrical pulses, you can generate the plasma inside. Once you've generated that plasma, you've ionized the gas, now you have to do something with it. And there are ways you can move that plasma with gas jets or with magnetic fields or with other kinds of systems. You can also use intensely pulsed magnetic fields to create glow discharges. You can use dielectric barriers to create a plasma. And then again, once you've generated it, you have to do something with it. Now in this schematic, you've got flat plate electrodes. The electrodes can be 2 plates, they can be a ball over a plate, they can be a needle inside a hole, they can be tubes next to each other. There's thousands of different kinds of electrode configurations. This is one of the configurations that we use in my laboratory. And I should say that most of the pictures that you'll see here are from work that's done in my group, just because I've got high-res photos of them and they're easy to get. So you can see on the bottom here, I don't know if my pointer is here we go. All right, so here you've got a couple of copper electrodes that are carefully shaped, and we've got about 40,000 volts that are put between those at about 3 millimeter spacing. So gas is coming in from the bottom, air. It takes that tight, hot, dense plasma spark and stretches it out so that arc glides up along those 2 pinning electrodes, which is why this is called a gliding arc. When it comes up and expands, by the time it's expanded up to the top, it's relatively cool, and you can actually put your hand on top of that, okay, without suffering, any kind of burn. And you get, again, free electrons. You get monatomic oxygen. You can have recombination to create nitrous oxides, and so on. So it's very effective. Now, I said this is a very tight spacing, 3 millimeters. Here we come to another kind of a system. This is a vacuum arc plasma system, also in my lab. This was built by my colleagues, Joe Seitz and Glenn Boyd. If you look at this, it's got kind of a similar appearance to the gliding arc system, but actually, because this is operating at a partial vacuum, you've got very, very different plasma chemistry that's going on in there. I don't have time to go into all the details about the reducting plasma chemistry, but when you're trying to compare multiple systems and say, well, I treated blueberries with cold plasma, you need to be very specific about using a common set of descriptors so that everybody knows what you're talking about and so you can compare one study versus another. That's important for the R&D phase where we're in right now, but it's also going to be important when you're talking about generating regulations to say what you can use and what you can't and what you can use that product or use that technology for. Right, so how do you make cold plasma? You can make this at a vacuum. The PD here refers to pressure and distance. So it's easier to generate a plasma if you've got lower pressure or if you move the electrodes closer together. All right? But the region that we tend to operate in our laboratory is around 1 atmosphere. And so if you change the gas composition of what you're putting across the electrodes, you can generate more plasma. All right? But some of these gases are going to be less effective at killing the bacteria or killing the viruses, and they might have different costs. Air is very cheap, helium is very expensive. So before you pick your gas, you got to understand how you're going to use this in an actual process application type system. So let's talk real quickly about the antimicrobial effects of cold plasma. And this is that same gliding arc plasma system that I showed you before, operating in a couple of different regimes. A, B, C, and D are nothing other than the amount of air that's flowing up from the bottom. That's either at 10 liters per minute, 20 liters per minute, 30 liters per minute, or 40 liters per minute. And the only effect that you have there is that the more air you push up through a rather carefully designed plenum at the bottom, is you're pushing that spark harder. So it forces it farther up the electrodes, reducing the time of flight between where the plasma is between the electrodes and where it's actually impinging on the either salmonella or the E. coli that I've put onto that golden delicious apple, all right? Reduced time of flight means that you have a kind of a statistical distribution of what kind of plasma reaction products are going on to the food, and therefore how effective the product is going to be. And as you can see for both the salmonella and the E. coli, this is time dependent. The longer you treat it, the better the kill you get, okay? We also did the same thing, this is with a different kind of a plasma jet system, inactivating Tulane virus and murine norovirus, which are surrogates for human norovirus. And I'll just point out here on this, as Nate said, some of these systems, they can actually give you quite a bit of a temperature input. So you say, well, is this truly a non-thermal process? On both of these, this is the temperature graph going up like that. You're going up to 70 degrees inside of 60 seconds. So what we did is we took that same system and we redesigned it and we introduced additional air jets to bring the temperature down to maintain it below the point where you would see thermal inactivation of the viruses, right? So this is staying here at about 45 degrees, a little bit lower than that in each of these cases, and you're still getting the same kind of inactivation for thermal or non-thermal, okay? So the antimicrobial effects, basically it's based on the oxygen singlets that are produced, it's based on the free electrons that are produced, and there are lots and lots of kinds of variations, which is a challenge for validation. The gas mixture that you use, I talked about, the pressure that you're using at, the equipment design, the shapes of your electrode, the spacing of your electrode, the polish of it, the composition of it, the applied voltages and frequencies, and it matters a lot whether you're using, say, a square wave or a sine wave or a spike ionization wave with some kind of a refractory period in between. The dielectric properties of the food that is being treated is also very important, as well as the physical properties of the food that's being treated. The same system can give you very different results with just one little twitch of the dial, and it can make kind of a challenge to say, my system is doing X, Y, and Z. You need to be sure that it's doing that all the time under those conditions. So this is another example of a different kind of a system where we're looking at UV production based on the humidity of the feed gas. Okay, where you take this from 5% humidity to 30% to 60% humidity, you can have a serious change in the characteristics of the UV spectrum that's being produced in this system. So you might say, well, my system uses air. Well, if you're not controlling the humidity of the feed gas that's going in, then you might be getting a very different kind of a reaction product than you're expecting. And it can lead to variations in your results, which might make it work on Tuesday but not on Thursday, which is a headache. But it also has some serious, impact in terms of your process validation later on if you're trying to run this for 1,000 hours on a production line. Just real quickly, the electrical properties of the foods. Here, again, this is some of the stuff that's from some work that we've done in my laboratory, past and present, where we're comparing, this is the same system here on cantaloupe versus eggs. The thing about the cantaloupe is it's got this netted surface, so you have a lot of turbulence right at the surface. So the way that the plasma is flowing around the surface trying to kill that salmonella is very different than the very smooth surface of the egg. Maybe you can see that where you've got this kind of laminar layer of dead air on the egg, which causes the plasma to slide around the side and interact less strongly with the egg surface, okay? And so you get a very different kind of a response when you're trying to kill bacteria that are on that. Here's a different kind of a system, a corona discharge system, killing seeds inside a container. Small, round seeds are going to behave differently when you're using the same system to kill salmonella on almonds, all right? Because the shape of the product is different, the configuration is different, and the way the plasma wraps around the product and the interaction of those the atomic oxygen and the electrons with the organisms on the surface is going to be very, very different. Another kind of a system over here, looking at blueberries inside a container, okay? If you don't have additional airflow to come in and move that around, then you can have a lot of heat buildup inside the container. Okay, so the shape of your container is going to matter, the composition of your container. Is it a flexible container? Does it allow for these things to move, or are they held rigidly? And if you're doing plasma for killing biofilms on foods and food contact surfaces, again, you need to talk about residence time. You need to talk about, are you combining this with other mechanical abrasion, scrubbing, and so on. All the plasma that I've talked about here is a completely waterless process. Okay, so there's no sanitizing solutions, there's no chlorine, there's just, air and electricity. But if you're going to be combining this with other kinds of sanitation systems, as you would in a production environment, you need to understand what impact these things are going to have when they interact with each other. Right, so just very quickly in conclusion, the term cold plasma, as I said, it represents a constellation of different kinds of processes, right? And they are related in terms of the underlying physics and in terms of the underlying ionization processes, but they are all very different. They need to be considered individually. A does not equal to B. They're all going to have lots and lots of different features in terms of their electrode design and their feed gases and the kinds of commodities they're going to be used for. So when you're talking about validating cold plasma, what exactly are you talking about? Okay, you need to give these details, and you need to have accurate, reproducible descriptions of your technology, the commodity that you're going to use it for, the treatment protocol, which how you're using it, and what are you using it for. All right, that's what the validation process has to be. Because ultimately you want to achieve the efficacy, the consistency, the reliability, and the traceability. You need to have do the record keeping on this to make sure that it's going to function the same way after it's been running for 1,000 hours as it did when you ran it for 5 minutes in the laboratory. Okay, and with that, I think I'm out of time, but I'll be happy to, take any questions. Thank you.
So how long does it take for these reactive species to combine? It's in the order of seconds, milliseconds?
The half-life on the monoatomic oxygen is real short. I mean, that's less than a second. But once that recombines, when the O2 cracks apart, you got these free electrons or monatomic oxygen. When that recombines, it'll recombine to O3. And that's got a longer residency time in the system and in the package. But like the free electrons, those don't hang around very long. You got very short half-lives on some of these things. And so the design of the systems are set up so that you are either putting the plasma right onto the food pretty quickly, or, because that can be very technically challenging, you just acknowledge you're going to lose most of those very reactive products, and you're going to rely on the secondary reaction products, and those are going to be the ones you can control and apply to the foods.
Thanks.
All right.
Hi. What is the safety, excuse my ignorance, what is the safety of plasma?
You mean to the workers around it?
Yeah.
Depending on the design of the equipment, it can generate some ozone. Some systems that I have in my lab, they don't generate very much. Others generate more. And it depends on, again, the type of the electrode and the shape of it and the voltages that you're using. It is 45,000 volts, so you don't want to go sticking your fingers into it. So compared to, let's say, other, high voltage industrial equipment, it's about the same.
Can I have another short question? Can you give some applications, although you mentioned a couple of things, applications that you've worked on?
Sure. Well, some of the stuff that I've published on has been to reduce biofilms on food contact type surfaces. And that's, we applying this plasma jet to Salmonella and E. coli biofilms, we were able to get better than 3 log reductions in 15 seconds for durable biofilms. We could see that there would be applications for sanitation in, like, say, pipes and drains, for continuous cleaning of conveyor belts. I just presented a poster, which anybody saw, I don't know if you did, on inactivating E. coli 157H7 inside a package on inoculated romaine lettuce. So we have a system where you can have the lettuce, put it inside a package, inside a conventional clamshell pack, put it into the plasma machine, turn it on for a while, and you knock down the E. coli. And that's, so that's, as a post-packaging process, we see that that could be incorporated along with all of the pre-packaging sanitation steps as an additional control level.
I was wondering, how would we measure the dose? Is it going to be ROS concentration? Is it going to be the intensity of light, or a combination?
I think it's going to be a combination. Probably, I mean, you could do headspace analysis to look at ozone concentration, concentration of other reactive products. One of the things you can do when you inject water vapor into the plasma is you break apart that H2O into H and O, and then that'll recombine to make H2O2. So you get hydrogen peroxide vapor as well, and that can be traceable. It does produce a little bit of UV, so you can measure the flux of that on the surface. And there, there would be just other, kind of chemical means to measure those, because mostly that's where the mode of action is going to be.
Rob Took, CDC, a question that also reflects my utter ignorance on the field, but the impact on a food, say, with specific qualities or viability issues, let's say seeds, disinfecting seeds.
Right. You're asking what is the effect of the viability?
Yeah, what does it do to the seed?
Well, I actually have some studies that are going on right now to suggest that treating the seeds actually enhances germination of seeds. So the project is to use plasma treatment of the seeds to knock down surface microbes as a safety treatment, but it also seems to be how about if I talk about other people that have published their work? They have shown that it enhances germination, and we're replicating some of that work. But we're also doing some other things, because when you generate this, it generates nitric oxide, and that has, can trigger in the pre-emergent embryo, it contribute some of the systemic induced resistance pathways in the plant. And what we're hoping to see is it will have physiological responses in the seeds after cotyledon expansion.
And if the seed happens to be a wheat seed, and if you make flour out of it, does it bake nicely?
Well, that's a good question. We haven't really tested that. I mean, our lab is pretty much focused on the food safety aspect of it, but,
This is food safety I'm talking about.
Oh, you're talking about as opposed to functionality of the starch.
Can't pasteurize flour if you can't bake with it.
Okay. You know what? Let's talk afterwards. We'll put together a project.
Okay.
All right. Sir, one more question, I'm told.
Yeah, just a question on the sensory side of things. Does it have any effect on sensory qualities such as taste and smell of, like, say, strawberries, raspberries? And is there any information on consumer perception of this?
Okay, well, I'll take the second part first. Consumer perception of the technology itself, no, not that I'm aware. Consumer perception of plasma-treated foods, no, not that I'm aware. This is still very, very early days for things like that. But in terms of what it does to the food product itself, the studies that we've done with lettuce show that if you keep the treatment at a moderate level, then you can have good color, good texture. We had, saw no changes in respiration, water loss, anything. So, turgor was good, texture was good, color was good. Taste testing would be the next phase of operations. When we tested those with blueberries and with tomatoes, what we saw is that if you really, really extended the treatments for a long time, you did start to see some sensory stuff. But before the sensory problems kicked in, you would still see, like multi-log reductions. So I think that, like with anything, there's still a lot of optimization that has to be done to get the kill level that you want without causing the sensory problems. There's going to be an optimized treatment that we're still working on developing. All right. Thank you.